going to talk about two important concepts in the role of how cells do some of their major processes inside of our body. And we're going to do a little bit of comparing and contrasting so that you can see how effective some of these processes are and what some of these processes do on a daily basis, if not on a second-by-second basis, inside of our cells, so that you can get a greater appreciation of all the hard work and energy that our cells end up accomplishing for ourselves and for our bodies. So we're going to be covering two objectives, which are both objectives that are on your notes that you're going to be following along as you're watching this video. So the first thing that we're going to do, we're going to look at two different processes. Some of them you've already are familiar with, and they go hand in hand with each other. We're going to look at diffusion versus osmosis. Now they're both going to have to deal with same things, both involving our cell membrane of our cells. Now if you remember from our organelle unit, a cell membrane lets certain things in and certain things out. It's kind of like the border patrol, as some of you guys put it in your cell projects. Or even like a fence or a gate. It allows some people to cross, some people not to cross. And this goes for the same for our cells. It allows some of our molecules to pass by easily, and some of our molecules not so much, because it's not needed inside of our cells at this time. And diffusion and osmosis is two processes that allow these things to happen. When talking about the cell membrane, there's a concept that is going to be relevant to this entire video, and that is going to be permeability. Some people call it semi-permeability, because what it does is it allows certain things to cross the membrane while other things are prevented from crossing the membrane. So it's like a locked door. Certain people have access and the keys to open that door. Other people do not, so they're kept out of that room. Same thing like the Border Patrol. Certain people are allowed into the country while other people are not allowed into the country. That's what selective permeability means, is to let some things into the cell and other things stay out of the cell. And that's what we're going to be looking at first when we're talking about diffusion and osmosis. Now with diffusion, we're just talking about normal small molecules crossing the cell membrane. Now to help me demonstrate this point, I have a screen, like you would see on like a window. The screen is full of holes. So you can probably hear that. That's the screen there. So the screen acts as a semi-permeable membrane. It allows other things through, but not all things. So what we're going to take a look at, we're going to use this screen as our model to help us figure out the differences between our diffusion and osmosis. Now diffusion and osmosis, there's only one thing that really separates them and makes them different. Diffusion deals with all small molecules. So everything that can easily fit through our membrane here, our screen. So that could be something like, salt. Salt is a very small molecule, to us at least as humans. So if I put a little bit of salt in my hands here, so you can see that it's salt. Nothing but the best. We can easily see that when I take the screen to it, and I throw the salt at the screen, the salt passes through the membrane there, because it's selectively permeable. The holes are big enough to let the salt go through. Same thing with our cells. The cell membrane has holes in it. Now these holes aren't called holes, they're called pores, just like your skin has pores. We use similar language for that. So the screen acted as a semi-permeable membrane to allow the salt to diffuse across the screen there. Now we get another aspect of osmosis. Why diffusion is different from osmosis is because diffusion deals with all kinds of small molecules. Osmosis deals with one important molecule, and that is the molecule of water, which I have in this jug right here. Now I'm going to simulate how some of these are the same by doing a little experiment here. I'm going to put the screen directly on top of our graduated cylinder here. Now it's acting as a selective permeable membrane, where the water is going to be able to go through the membrane to the other side. How diffusion and osmosis both work is that they go from areas of a higher concentration to an area of a lower concentration. Essentially what is going to be happening is that the cell is always looking to keep itself in balance. We talked about before the word homeostasis, making sure we have stable conditions. Diffusion and osmosis help us maintain homeostasis for our entire body. So if we take our example here, we have this whole 2-liter jug of water. It's full to the 2-liter mark. About 2 liters of water in here. This graduated cylinder measures a liter of water. Let's say that the graduated cylinder is representing our cell in this case. The cell needs to be balanced with the outside environment. So what would need to happen is that the water from the outside would travel into the cell because it goes from an area of high concentration, the jug is full, to an area of low concentration. The graduated cylinder is not full. So if we pour through the membrane, it passes through the membrane easily. Because it's water, it's a small molecule. H2O, it's made out of hydrogen, 2 parts hydrogen, 1 part oxygen. All are very small molecules, comparatively speaking, on our periodic table. Now, at the point where we get about balance there, there's the same amount of liquid both in the graduated cylinder and in the 2-liter bottle there. This would mean that it would be a happy cell at this point because it's maintained its homeostasis. It's balanced both outside and inside. Whenever things end up changing, the levels have to adjust with it. If there's ever more water on the outside, the water wants to move into the cells. Whenever there's more water inside of the cells, the water wants to move outside. But some of the facts that go along with it is that this doesn't require any energy on the part of the cell or even our body. This is something that just happens naturally because the body wants to take care of itself and balance things out. So there's no energy involved with this. So we're not wasting our hard-earned energy that we get from eating food and getting all the necessary molecules that we need to carry out our day-to-day basis. It doesn't require any extra energy on our part. All that's required is for our cells to realize that there's an imbalance in their molecules. And it transports from one area to the next. Here's going to be the first set of notes there. I'll try to put it as close to the screen as possible. Hopefully I wrote big enough so that you can read it. If you need to, pause the screen now in order to copy all of the rest of the notes. Alright, in our second part of our video right now, we're going to take a look at what happens in 3 different types of cells and how diffusion and osmosis work. Now, this follows the same type of pattern that's on your page as well. We have our normal cell, too much water inside the cell in the middle, and then too much water on the outside. And we're going to see and find out what happens to each cell as diffusion and osmosis takes place. Our normal cell is going to do everything that we expect a cell to do. Water is going to be, which I'm going to be representing with my blue marker here, water is going to be traveling out at the same rate as it is traveling inside. There is no change in the size of the cell underneath normal conditions. Water is going to be leaving and exiting at the same rate because it's already at that stage of homeostasis, so no other actions need to take place. Now things change whenever we get an imbalance of our conditions. If there's too much water inside of the cell itself, what's going to happen is that there's a whole bunch of water molecules that is inside of the cell, and maybe not so much on the outside. So you can see that there's more inside than there are outside. What's going to happen if osmosis is going to take place is that the water is going to be moving from the inside to the outside of the cell. In these conditions, this would cause the cell to shrink in size and get smaller because the water is physically leaving the cell. And when the water is physically leaving the cell, it's going to make the cell a lot smaller. So it's going to shrink. Now for our final one, we're looking at too much water on the outside. So it's the reverse conditions. So most of you probably already know what's going to happen. Since it's the reverse conditions, we're going to have very few molecules on the inside, but then it is covered on the outside with water. So there's water everywhere on the outside. There's not that much water, comparatively speaking, on the inside. So what's going to happen is that the water is going to go from the outside of the cell, and it's going to work its way into the cell. So it's the reverse process. So instead of shrinking, these cells are going to grow. Now this is the same thing that happens to you when you're in the bathtub for too long or in a swimming pool for too long. And you notice that your hands get wrinkly. The wrinkling is actually because your cells are growing and it's causing them to be inflated with water. That's why when you dry off, they start to go back down after a while because you're getting rid of all that imbalance of the water. Your skin is actually absorbing some of that extra water, which is causing your cells to grow. The wrinkle shape is because your skin is attached to your muscles at certain locations, so that creates the wrinkling effect of your skin. But the main point is that they grow because of the influx of water. More water is going into the cell in that example there. If you haven't followed along and copied down what the cells are, this is in the middle of the page of your notes. Go ahead and pause this video to make sure that you copy down everything that you see on the board in front of you for this video. Alright, in the last part of our video today, we're going to look at active and passive transport. Now this is something that we've already kind of hit on before, because passive transport is just the general term that we can call it when things like diffusion and osmosis happen in the cell. What passive transport is that it's transporting molecules from one location to the other without the use of energy. So it's just something that happens naturally. Something else that goes along with passive transport, as we looked at with diffusion and osmosis earlier in the video, is that these only occur when things are moving from a higher concentration to a lower concentration. So it's like if you have a boulder on top of the hill and you roll it down the hill, it's naturally going to want to go down the hill because of gravity. Things are naturally wanting to go from high concentrations to low concentrations, which means if you got a lot of something and a little bit of something, they're going to start to even themselves out. So that's what passive transport is. That's what we've talked about. What we haven't mentioned is active transport. And active transport is going to be a little bit different because it's going to require the use of energy and harnessing that energy to allow bigger things to cross the cell membrane. Because as we mentioned before in the video, with our cell membrane, it's like this screen here. It's got holes, but the holes are only so big. So it can only let certain molecules through. Some of the molecules that we need on a daily basis are bigger than what our membrane can support. So we have to figure a way to get that through our cell membrane. Things is energy. It's the use of energy. It's active. Just like when you're active in anything, you're using your energy. Instead of passive, you're kind of laying back. So the words themselves help us think of and remember what active versus passive transport is. Now, a way to demonstrate this is that, let's say we have one of our larger molecules here, which is this kind of wiffle, golf ball, whatever you want to call it. This is too big to fit through the screen. It bounces off. In the cell, that's what would happen. It would just bounce off. The molecule would try to get through, and nothing would happen. It would just keep on bouncing off. No matter how many they brought to the table, end up bouncing off. So that's what just happens for active and passive transport. Now what we end up doing is that because we use energy, we're allowed to alter the situation. What happens is that the membrane is covered in proteins. These proteins act as tunnels that allow the molecules to pass only if they're of a certain size. There are hundreds and thousands of proteins on the outside of our cells which recognize different substances. Each one of those proteins is like a key. The keys only fit a certain lock. So when the molecules come and they want to pass through, they must have the same shape as the protein, and it unlocks the door to allow them inside. And that's just what happens during active transport. Now the reasoning for why active transport needs to happen is because our cells use a lot of different components on its day-to-day basis. So it needs more molecules inside the cell than what is outside. So typically this works backwards from our passive transport, where things are moving from high concentrations to lower concentrations to balance things out. Active transport is doing the complete opposite. It is taking an already higher situation and adding to it and making it greater and greater and greater, because the cell is constantly using those resources. So it's going to constantly need those available so that the cell can do its daily functions when it needs to. So it typically works backwards from our passive transport. Now, the one thing that brings all these together is the fact that both of them are used to transport substances into and out of the cell. And the cell determines what is allowed to go into and out of the cell. It's that selective permeability. It allows things in, but it keeps other things out. That finishes our video for today. Notes, so there's the final remainder of the notes there.